Hello guys. In the earlier chapter, you studied about DNA. What are DNA? Their basic molecules. What and all there in the DNA. Then we did not study much about RNA. There's the RNA is another major molecule. But when we talk about a DNA, the structure of DNA is so important. We studied what are nucleotides, what are nucleosides, what are the sugars present, what is phosphate, all those things, and the bases too, nitrogenous bases too. But we did not concentrate much on a model of DNA. But if you look at the history, there was no in before 1950 there was no clear idea of what is the structure of DNA or what was the structure of DNA. So then Maurice Wilson and Rosalind Franklin, they were studying all the biological molecule with the help of X-ray. You know, right? When we apply X-ray, it will diffract from that, and by using the diffracted pattern, you can somehow study or assume the structure of any molecule, any biological molecule, and with that we can elucidate the structure. And in 1953, as we all hope, in 1953, Francis Crick and James D. Watson, they took this data from them and they elucidated structure, and later they got a Nobel Prize for their work. So according to Watson and Crick, what is the structure of DNA? But when before studying the structure of DNA with the help of Watson and Crick, there was a lots of controversies with regards to the structure of a DNA, because there are lots of reports that Rosalind Franklin should be given the credit for the structure, but the real in the what is literature available, the Watson and Crick got a Nobel Prize for this work. So according to Watson and Crick, what is the model of DNA? So DNA consists of two anti-parallel chains. The two anti-parallel chains means the one chain is running from here and ending here, another is starting from here and ending here. So that means they are anti-parallel in nature. They are helical in structure, they are anti-parallel in nature. There is two important things, helical and anti-parallel. And if you see this helical backbone, it is made up of sugar and phosphate. Sugar and phosphate is the major molecule which is holding entire backbone of DNA. Then we have a stacked bases inside that. There are bases like adenine, guanine, cytosine and thymine. And there are sugar which is called which is there in the backbone is deoxyribose sugar. It is in DNA, and if it is RNA, then we call that as a ribose sugar. So these adenine, guanine, cytosine, all these bases are stacked one over the another and form the entire structure of DNA. And how these bases are held together? These bases are held together by what you call a hydrogen bond. Adenine is always bond with a thymine with the help of two hydrogen bond. And if it is guanine, then guanine with cytosine with the help of three bond. That means if adenines are more, the holding strength may be less. If it is guanine, then the holding strength is more. So because the three hydrogen bonds are, really speaking, hydrogen bonds are weak. But if you put lots of hydrogen bonds together, it gives a very stable structure. The same thing is happened in case of DNA. DNA double helix are so stable because of its large quantity or large number of hydrogen bonds. So if you see the structure, there are major grooves. You can see it here. There are minor grooves. That means in a three-dimensional space, when you arrange any spiral structure, there are grooves of form. One is called a major groove, another is called a minor groove. If you see the base, stack base, there you can see the distance between the base is here. I have mentioned that is 0.3 nanometer. And if you see the one helical turn, it is approximately 10 bases are there. If you count there are 10 bases, that way it is 3.5. But if you see the entire diameter of this, that is around 2 nanometer or 20 angstrom. But you just look at the structure. There are helical the helical strands are there which are running anti-parallel in nature. There are bases which are stacked one over the another in a specific distance. They are holded together with the help of hydrogen bonds. But if you look at the structure, it is entire thing is coiled around a single axis. The entire structure is piled around a single axis, which is giving a stability for DNA. That's why this proposed model of DNA is well accepted in the scientific community, and later that's why they awarded Nobel Prize for this simple what you call a very big contribution to the field of molecular biology. Let us move a little more further than this DNA model. So there is another concept which will come in the DNA, that is called hyperchromicity. What is the meaning of hyperchromacity? You know that the DNA is always found in double stranded structure. If you convert this DNA from double stranded into single stranded DNA, that means DS, double stranded DNA into single stranded DNA, then the absorption of UV light will increase. You might be thinking, why this UV light absorption is increasing? It is increasing because when you increase the surface area, the absorption will increase. That means same thing. If you smaller the particle, the surface to volume ratio is high. So what happened? It absorb more, and this increasing absorption by any material, then we call that phenomenon as hyperchromacity. So hyperchromacity is the increase of absorbance of a material. Here it is a DNA. The hyperchromacity of DNA that occurs when the DNA complex is denatured. The UV absorption is increased when the two single-stranded DNA are being separated by heat or by addition of denaturant or by increasing pH level. Anything you can do, you can denature them and you can get the hyperchromacity. That's the concept of hyperchromacity. So what is responsible for this hyperchromacity? That's a melting. So DNA has to melt. That means the double stranded DNA has to convert into a single stranded DNA. If it is a double stranded DNA, it has to convert into two single stranded DNA. So once it is converted into, around 50% of the DNA, if it is converted into single stranded DNA, then we call that particular temperature is called as a melting temperature. If a GC content, that is guanine cytosine content, are more, its melting temperatures are high. If it is AT content, then melting temperatures are low. It is mainly because GC are whole two. That means guanine cytosine consists of 3 hydrogen bonds. That means more energy is needed to break that. And if it is AT, it is 2 double bonds. So less energy sufficient to break this entire thing. So initially we studied Watson-Crick double stranded model. Then we studied what is hyperchromacity, that is increasing absorption of UV light by converting double stranded DNA into single stranded. Then what is responsible for that? That is a melting. Melting means at this temperature, the 50% of the double stranded DNA will be converted into a single stranded DNA. It is completely depends on GC content and AT content. If it is GC more, high temperature is needed. If it is GC less, the AT rich, then the low temperature is sufficient. It is even represented in the graph 2. Let us move on to another concept, another simple concept in molecular biology, that is called as Scott curve analysis. This is very significant, very essential. Why? This Scott curve analysis will give us a clear idea of what is the complexity of DNA. Whether DNA is very complex DNA or whether DNA is a very simple DNA. What is the nature of sequences? Whether there are any repetitive sequences are there. If it is repetitive sequence, whether it is a GC rich sequence or whether it is a AT rich sequence. So it's the entire clear idea of what is the complexity of DNA. So this is a simple technique developed in 1960 by Britton and Davidson. So this entire process is completely based on DNA denaturation. You might be thinking now, what is denaturation? Because already you studied what is denaturation. Denaturation means a double stranded DNA converting into single stranded DNA. Initially it will be like this. So now it is like this, single stranded DNA. Initially double strand, then it converted into single strand. So the Scott curve analysis is completely dependent on this double strand, single strand nature. So what is the principle of this? The rate of renaturation is directly proportional to the number of times the sequences are present in the genome. If repetitive sequences are more, it can renature faster. That means anneal fast. They can join fast. Given enough time, all DNA that is denatured will reassociate in a given DNA sample. That means if you give it a time, everything will renature and form normal DNA. All denatured DNA will be renatured back to the normal DNA. The more the repetitive sequence, the less will be the time taken for renaturations. That means renaturation is completely depends on the sequence of repetition. And if you give a time, all the DNA will be renatured. And the time taken by the DNA for renaturation is completely depends on repetitive sequences. So how to study this? The process involves denaturation of DNA either by heat or by chemicals and allow them to re-anneal by cooling. The renaturation of DNA is assessed stereophile. Large DNA molecule takes longer time to re-anneal. That means if a molecule is very large, it takes longer time to join. If it is a simple single copy DNA, it won't get what you call renatured. So the time, anyhow, the time is less. If a smaller DNA, time is less. If it's a bigger DNA, the time is needed more, because it has to, whatever thing you unzip, you have to zip it back. So unzipping means the opening all hydrogen bonds. Then this hydrogen bond has to form, so time is needed more in case of a double stranded very lengthy DNA. So you just have a look on this COT curve. So here percentage of remaining single strand DNA, then COT values are there. If you see this, you can see the 100% highly repetitive contents. That means if your repetitive contents are more, it can form 100% double stranded DNA. Then if it is a middle repetitive, then the single copy chances are very less because there is no double repetitive. If it is repetitive sequences also, it cannot. So highly repetitive, middle repetitive, and single copy, a single fragment. I mean the chances are very less because attraction may be very less between those two renatured fragments of DNA. So let us move on to the last stage, that is the significance. What is the significance of this COT curve analysis? So you can understand the genome size. What is the genome size and what is the complexity of a genome, whether it's a highly complex or it's a simple genome. Understanding complexity of sequences. One is a genome complexity you are understanding. Second thing, you are understanding a complexity of sequences. Then finally, the understanding relative proportions of single copy and repetitive sequences. How many single copy are there? How many repetitive sequences are there? That means a genome size, complexity, sequence complexity, single copy, and repetitive nature. How many idea or many things you can learn just by analyzing a COT curve or COT value of a particular DNA fragments. So this entire process is completely depends on renaturation. Not on the denaturation, but it is on the renaturation. So the entire chapters we studied what is the structure of DNA. That is a double helical structure proposed by Watson and Crick. They got a Nobel Prize for that work. Then we studied what is hyperchromicity, increasing the absorption. Then we studied TM, that is a melting at what temperature the 50% of double stranded DNA will be converted into single stranded. It is completely depends on guanine cytosine content than a adenine thymine content. If it is guanine cytosine content or more, it takes more time or more temperature is needed. Then we studied COT analysis. It is clearly measuring the complexity. It depends on renaturation. Thank you.